There's a reason we have this order of draw. The reason is because of the additives that are in these vacuum tubes. Okay, so my yellow, which I don't have here on this table, red, light blue, these things that's inside of them that gives the blood its properties for whatever sample state that we want to test will interact with each other if one is mixed with the other. So if we do one first, okay, then we don't have to worry about this one interacting with this one if it comes after. If we did it first, there would be a problem. There would be error results or the samples would be tainted. So we have to make sure we do some after others. Okay, and then also each of these tubes stand for different types of tests. We're going to review that today. And I think I'm going to start right here on page 127, and then I will go back and cover the previous part of this chapter on Thursday. We'll finish this chapter up. Just because it's so crucial and I got everybody here. So starting at the bottom of page 126, it says the types of blood specimens. Can anybody tell me what those are, just right offhand, other than Marissa? Actually, Marissa, go ahead and tell me. Plasma. How do we get these three? How do we separate blood into one of these three components to test it, right? The whole blood is blood collected, I'm reading the top of 127, and mixed with an anticoagulant so that will not clot. Okay? So I'm gonna go ahead and put that right there. Anticoagulant. Hannah, what does that mean to be against coagulating, anticoagulant? It's not going to clot. In other words, your blood is going to stay in the whole form, just like it came out of your arm into the tube. It's going to stay just like that because there's a chemical in there that's going to keep that blood from activating clot factors and stuff like that. This is good for when we do test any hematology test, like cell counts, complete blood counts. I'm going to put shapes here, but the real word is morphology. Okay, are those cells being produced correctly? Are they sick, like they have sickle cell shape or something? We want to make sure they're formed correctly. If you want to look at any hormones in your blood, the paragraph also states, well, that's pretty much it. Metals. So we might have to worry about lead poisoning, or we want to make sure you got a certain metal like magnesium or iron. We need whole blood for that. Okay. There are three types of whole blood, this is one part that you all are in the dark about. Marissa was here for. We've got venous blood, arterial blood, and capillary blood. Three types, okay? Tell me a little bit about something about venous blood, Marissa. It's in my veins, first of all, right? Not my arteries. What's one characteristic of this blood? We color our veins blue or red? Blue. Veins have blood that is low in oxygen. That's the only place in the body where it is like that because it's within the pulmonary circuit. Everywhere else in the body where we're drawing blood, you can kind of put that to the side now and think about blood you're drawing out of people at this point. And if I draw blood out of your vein, that vein is on its way back to the heart for that blood to be clean and circulated back throughout your body. You call pulmonary veins red because it's still technically draining towards the heart. It's still a vein, but it happens to be coming from the lungs and it's oxygenated. It's flip-flopped in this one area. And then the arteries are also flip-flopped in that one area because they're carrying blood from the heart to the lungs, but it's blood that's on its way to be cleaned. That's the only place you have to worry about that. And you can almost kind of forget about it at this point, because now we're talking about blood we draw to people from here on out in the course. All right, so this is low in oxygen. It's typically considered blue in our books and in our diagrams. And when we look at blood in your veins, you can see your veins are sort of blue. That's because light is reflecting off of that blood. Okay, it's low in oxygen, and that's just the color it gives us. We turn blue when we lose oxygen, right? When we lose our breath, same thing, okay? So low in O2. Arterial blood, on the other hand, is bright red. It's in our arteries, and it's under a lot of pressure. Think of the place you can feel your radial pulse. That's the radial artery. It's strong enough where you can actually palpate it when you squish it up against the bone. You can't do that with veins. Pressure's not high enough. Okay. High in oxygen, other arterial gases we can measure here. We'll talk about one time we may draw from arterial blood. It's rare for a phlebotomist, but it's special training, but it's a possibility. Later in the chapter 13, I think. And then capillary blood is right in the middle. It does resemble arterial blood a little bit more, but capillary blood is between clean and dirty. It's where the exchange with the cells takes place in terms of gas, nutrients, whatever it is blood needs to do for cells. Okay, capillary blood is what you would test from a finger because that's not a vein or an artery, it's a capillary, microscopic, right? Okay. So you get why we need to keep blood whole. There's 3 types of that. The second form of this fluid we take out of our bodies, I'm going to rearrange this here real quick. Plasma. Plasma is the fluid portion of blood, including fibrinogen and other clotting factors. So I'm going to put that first thing. All right. Well, you're right. I'm just going to make sure she can still hear me.
Yeah.
Can you still hear me, Haley?
Yeah.
Okay, I was just checking in on. They must be having a fiesta or something in their house.
It's a what?
Why is that important? What's significant about clotting factors? They help us to do what? Clot. They help us keep our blood in our body, right? Can you give me one clotting factor? Do you know? Look, you might want to go back to chapter 7 and look at that clotting cascade. Things like fibrinogen and thrombin, they're going to be in plasma. Look at the bot right there. You were on it. Page 112. So things like prothrombin and fibrinogen. If we separate plasma from whole blood, then these are still going to be here. Okay. And sometimes we want to make tests without that. So we have to take them out. And that leaves us with serum. And it is just plasma without clotting factors. This is a very important concept. I want to make sure you get. It's like one of the first things we ever talked about. Giving up on that. Okay. So how do we get clotting factors out of serum? That's what we're going to learn here in a minute. Okay. It's really got a lot to do with that composition of blood thing we're talking about. So these are the three things we can test. And you know how we've got some tubes that say serum, some tubes that say plasma, and the lavender tubes say whole blood on them. That's what we're talking about. Okay. So plasma, just reading here, number 2 on 127, is the fluid portion of blood, including fibrinogen and other clotting factors. Plasma is obtained by collecting whole blood in a tube containing anticoagulants that prevent clotting. The sample is then centrifuged and the plasma is aspirated or sucked out of the top of a little syringe or tube. Okay, plasma is used for coagulation studies. It is also used for short turnaround time stat chemistry tests where there is no time to wait for clotting to occur before we spin it down or centrifuge it. Okay? So if I want to know how you're clotting, I'm going to need this. I don't need all those other cells that we could look at here. I only need this stuff. All right. And this takes time for this to clot up and sink to the bottom where I would have serum afterwards. So if I don't have time for this to do a chemistry test, I can use this, even though it's not ideal. My machines like to have pure serum that's got none of that stuff in it. But that's what they're talking about. And so serum is the fluid portion of blood that remains after clotting. The clot traps cells, platelets, and clotting factors. No anticoagulant is used collecting a serum sample. So if everybody remembers, we've had a blood or I've had a tube of blood before that we filled up with regular blood and we let it set and clot. And after that was done, there was a yellow straw colored fluid up here. And there was a dark red clump down here. Okay, this was all the cells and clotting factors where they had been activated and clotted and sank to the bottom. So if I take out clotting factors, what do I have left over? Serum. Okay. And once that's done, once we've given it 30 to 60 minutes to clot, then we can spin it down and separate any minute leftovers and get that sample really pure. And we can take it out and do our chemistry test or whatever else we want to do. All right. Unlike plasma, serum does not contain fibrinogen or other clotting factors, which are left behind or used up in forming the clot, and thus cannot be used for routine coagulation studies. Complete clotting takes 30 to 60 minutes, depending on the presence of clot activators. During that time, the tube should remain in an upright position. Leaving the tube upright during clotting prevents adherence to the sides and the stopper. So if you take your tube after filling it out, you might invert it or whatever, and you just lay it down on the table like that, then the clot's going to form here. You don't have that nice column of separation where you can get your sample out. So they're saying, don't leave it like this. Don't leave it like this. You want to leave it upright. So we got those purple things back there. When this occurs, residual cells in the clot can contaminate the serum, resulting in erroneous results. Once it's fully formed, the sample can be centrifuged or spun down to separate the serum from the clot. It's important to allow the clot to form fully, otherwise strands of fibrin in the serum will interfere with chemistry instruments, causing bad results. Okay, additives. Most blood collection tubes contain one or more additives. Additives include anticoagulants to prevent clotting, clot activators to promote it, and thixotropic gel to separate components. Don't be afraid of that word. It's just an inert looking snot stuff in the bottom of this. It's just like gelatin. Nothing special about it. Any tube containing an additive must be inverted and mixed well immediately after removal from the adapter. Gently turning the tube over, then back again, that equals 1 inversion. So 1, 2 is what they're saying. You should never shake a tube because it will cause hemolysis. What's hemolysis, Hannah? Yes, very good. The exact number of inversions needed varies by the tube type, but most need 5 to 8. Okay, so I'm gonna erase this if everybody's good with it. And we're going to talk about the different additives that we can use to get one of these 3 samples. And we might have to go over this again. I don't know if I'll even get to most of it today. As a matter of fact, I want to, I've got a paper that has explained out a little bit better. I made a long time ago. Let's see if I got copies of it. I'll probably have to search for it on my computer. I'll have that for you on Thursday for sure. Okay, so anticoagulants. They prevent blood from clotting. The first one, sodium or potassium EDTA, ethylene diamine tetraacetic acid, binds calcium, therefore inhibiting coagulation cascade. Go back and look at page 112 again. Find calcium, which is denoted as Ca in there, or Ca2. See where calcium is? It binds it up. That way you can't clot. Takes that out of the equation, so no clot can't form. Other additives that bind calcium include sodium citrate and potassium oxalate. Okay. And sodium, I'm just going to put SPS. Put SPS here. And then finally, the last one's heparin. That's our 5 main anticoagulants. Okay, heparin can come in 3 different forms. It can come as heparin sodium, heparin lithium, or heparin ammonium. I'll show you. We'll make sense of this in a minute. It inhibits clotting by preventing conversion of prothrombin to thrombin. Go back to 112. I'm going to go down here under the common pathway and see where prothrombin is activated into thrombin. That's where heparin works at. Pretty neat, huh? These tubes that contain this anticoagulant have a precise amount in them. You can see this light blue in here. It has a little bit of liquid in the bottom. All right, and that is sodium citrate. So you have to make sure you get enough blood in here to spread this anticoagulant out. If there's not enough blood to anticoagulant ratio, it'll mess your results up. So these have to be filled up all the way. And technically, if you can put 10 parts of blood in here, this would be 1. So it's 9 to 1 in this tube, which is a test question, hint, hint, hint. Okay. You gotta make sure you fill them up completely and mix them well. Can be over diluted or anything like that. What could happen is you could get tiny microclots in these tubes, and then that could gunk up your machines. One thing. All right. Which one of these do we use? How do we know what to use? Well, it depends on what test we want to look at. Okay, EDTA for whole blood is good because it doesn't affect red blood cell shape too much. It's good for that. It preserves blood cell integrity well. It also prevents platelets from clumping. Do you remember in the clotting or the stages of hemostasis, there was adhesion and aggregation of platelets? It's going to prevent them from sticking together. And it's compatible with blood staining, which is when they'll stain your cells with some kind of pink or purple dye, and you can look at it under a microscope. Otherwise, cells are clear and translucent. You can't make heads or tails of them. So we have to stain them, and this doesn't mess with it. Look on the next page and tell me the first tube you can find that has, on page 129, which one has EDTA in it? Give me hints on page 129. Okay with stains. That C with a hashtag means with. And actually that means without too, so. Okay, so it's purple. My question that I asked about which tube contains it, lavender does. It's the last one in our blood draw, right? So that means that EDTA must really affect other additives or not, or if it comes last in order of draw. It does. So they're going to put it at the end. They don't want it interacting with anything else. But it's still pretty popular, pretty good one. The next one, I'm reading at the bottom right side of 27. This is all like 1 or 2 paragraphs. I'm just really breaking it apart. Okay, EDTA interferes with coagulation studies. So we can't use it before we do a blue tube. Citrate is used for coagulation studies. Okay, and if this tube right here, light blue, is for coagulation studies, that means I'm going to have citrate in it. Okay, blue is for coagulation studies, so it contains citrate. SPS is special because it's used for blood cultures. It inhibits certain immune system cells. And that's important because when I draw blood to see if I've got some bacteria working in there to make me sick, I want that blood to be in the same state it was at the moment I took it out of my arm versus the moment it's put in the machine to analyze. So I don't want my immune system continuing to kill any bacteria from the moment it's drawn until the time I get it back out to the lab. I need to inhibit that process, and that's what SPS does. Okay, this is for blood cultures. Everybody follow me so far? Okay. Haley, you still follow me there?
Yeah.
Okay. SPS comes in this tube right here. The reason I don't have any blood culture tubes out here is because they're a little bit special. They look like, okay, so they're right here. These. This is way different from one of these little tubes. Okay, I can look on here, it says Bac-T alert, aerobic pediatric. Didn't tell me the additive exactly that's in it, but it's something like SPS. So if I'm drawing blood into this jar to see if I'm getting bacteria, it's really easy to contaminate this sample with bacteria that might come from my skin, come from an unwashed hand, come from something I touched inadvertently. So I have to make this the first draw. That way I'm not drawing these in this and possibly contaminating it with something that got into there. So when you draw a blood culture, you're actually sterilizing the site with iodine, and you're drawing two of these tubes, which we'll talk about later, why you're drawing two. It's always first, so you prevent any contamination from this other work that you may have to do, all right? Remember, these things we use to draw blood like these butterflies and these vacuum tube holders, these are multi-sample systems. So we're sticking tubes in there. We need to do it once for the blood culture and be done with that. Then we can go forth with other tests. So that's why it's first. Capiche? Okay. Moving on here. It also neutralizes antibiotics. I'll put that up there. That stands for antibiotics. Neutralizes antibiotics. Okay. Heparin is good for blood chemistry. Things like sodium, potassium, different types of gases in our blood, CO2, oxygen. Okay, and heparin goes right here in green. The last one right now is potassium oxalate. It's cool because combined with something else called sodium fluoride, it is used for glucose determination. Okay. Let's go over here and look at page 129 under the gray tube. The gray tube picture is at the bottom of page 129. There's a paragraph explaining it. It's at the top. Okay, for lactic acid measurements, glucose tolerance tests, which is what Haley just had, fasting blood sugars and blood alcohol levels. So if you have a wreck and you're in the ER and I think you've been drinking, I'm going to use this tube to test your blood. Okay, sodium fluoride is an anti-glycotic. What is this right here? Anybody? It's sugar that we need for fuel for our cells. Okay, the process of breaking that down and using it is called glycolysis. Looky there, we're breaking apart glucose molecules. And if we want to inhibit this, because I want to know how much actual glucose you have in your blood, remember, stuff is still going to be working from the time we put it in the tube till the time we get in the lab. We have to stop it at that point. Then we're going to use something that inhibits that, an anti-glycolytic. It's just going to inhibit glycolysis. Okay, don't forget, you may add or not add this sodium fluoride right here. I'm not going to spell it out. Okay. And so if we're going to use a serum sample, you're only going to have sodium fluoride. If you have a plasma sample, it's going to be either potassium oxalate or EDTA, in addition to NaFl. So you can use plasma or serum. That just gives us flexibility. If I draw one, say, oh, I forgot I need to add this glucose test to my panel, you can do that pretty much in any scenario here, okay, if it's not been too long. So gray, we've got potassium oxalate with or without sodium fluoride. Okay, so these are all anticoagulants. Notice we've got a gap for two colors here still. What are those two colors used for? Which sample? Red and gold. Serum. Looking right here under red, it says chemistry, serology, blood bank, specimen serum. I can look under gold Hemogard or the tiger top, red, gray, serum. And so if I want to take the clotting factors out of plasma to equal serum, that means I can't have something that's going to inhibit that. All right. And as a matter of fact, I want a clot to form to get serum. So what kind of additives are going to be in these two, Marissa? Or clot activators? Haley, do you need more time writing here? Are you taking notes?
I'm taking note and I'm reading it.
Okay, I can take a snapshot of this if you need it later. Fill in your notes.
Yeah.
I'm going too fast.
That'll work too. Make sure I got everything written down right.
Okay, I'll slow down a little bit.
All right, thank you.
So now you should be able to at least line up a few tests, or you're getting to the point where you can start lining up a few tests with each color here. Okay, I'm going to turn the page, look at 128. Just to recap about glycolysis, there's an important paragraph here that explains it. Sodium fluoride inhibits glycolysis and can be used for glucose determination. Glycolysis is a cellular reaction used to harvest energy from glucose. So potassium oxalate and sodium fluoride is used for glucose determination. All tubes containing anticoagulants should be gently inverted 5 to 8 times directly upon collection. Right. Clot activators, that's our other additive. These things promote coagulation. They also have to be inverted. They facilitate a fast clot. Okay, we don't have time to wait 60 minutes, which is the natural time it takes for our blood to clot in a tube on a bench top. With these additives, it takes only about 30 minutes. It turns that time in half. Thrombin is one of them. Okay, so I'll just make a little space here. And if you go back and look again on page 112, I see thrombin down here in the common pathway. After prothrombin, it also activates fibrinogen. Thrombin is a natural clot activator that we've taken and synthesized and put in the tubes to also promote clotting. Okay, but all kinds of things can be used as clot activators. Inert substances such as glass or silica that can promote clotting by providing more surface area for platelets to activate. Remember, platelets, once they start swirling around something or getting close together, they start clumping and activating. So if I put them in a glass tube, that if I zoomed in here with a microscope and looked at the surface of the inside of this tube, it would be really rough and jagged for glass. And that will cause platelets to start clotting as they are just sloshed around in there. Okay. We can also put stuff in this tube like sand or clay that will also cause those things to bump around and start to clot up. Just remember, you gotta invert these two. Any questions about that? Clot activators are pretty easy. Okay. Thrombin's a major one. We don't like to use glass tubes anymore because why? They can break, and if they break, they can also cut us. And if they have blood in them, that's bad news for us, right? We don't want to be changing blood out people. I'm not going to write the gel up here. It's a little bit too much, I think. But it is an additive in tubes in the sense that it's been put into some of these tubes. It's an inert substance whose density is between that of cells and that of blood serum or plasma. All right. When this blood is spun in that centrifuge, or when this tube is, this gel becomes liquid basically. And when we stop spinning, it will solidify back. And so red blood cells are heavier than this gel when it's liquid, and they will sink through it to the bottom. But serum is lighter and it will stay on top. Okay, and keeping all of our chemistry stuff with it. And so when it stops spinning, it will gel back up, and we have a nice separation of two layers, serum and a clot or whole parts at the bottom. We can draw that out and use it to test really good, some chemistry stuff without messing up our machines. In light green top tubes used for determining potassium, the gel prevents the plasma from being contaminated by potassium released from red blood cells. I would highlight that sentence at the bottom of polymer gel. One side effect of red blood cells being broken apart is the release of their contents, and specifically potassium. And so you can infer that if there's extra potassium in my blood sample, I might have false elevated potassium levels, right? So we don't want to hemolyze our cells. Thankfully, whoever invented this vacuum tube system and this multi-sample needle container, color coded it, made it pretty much easy, okay? Almost foolproof. You just have to come to school here to be able to learn how to use it. Any questions about that? Nope, nope, nope. Okay. So at this point, Haley, the beginning of the chapter is a lot about stuff that you've already been told about.
Yeah.
Like, the needle, the lumen, the bevel, okay, the hub that we attach to it, the little rubber end on the inside that goes into the tube and creates a seal,
Mm-hmm.
safety device that covers the needle up after it's been used. Okay. That's what the first part of this chapter kind of describes. Did I ask you all to read this before? Didn't I ask you to read this first part of this chapter way back on like the first day? You ready for blood draw? I don't think that was this class. One class I said, I want you to go straight to chapter 8 and read that chapter first. Because I knew the next day they were here, they would be drawing blood and I wanted to have an idea about it. So I'm going to leave that up to you. I'll review with you all later, but that's something you can read it on your own and kind of know already what you're reading. All right. So at this point, I'm going to say, Haley, you're good for the most important stuff. And I can turn the camera off and let you go, and then we can cover the rest later. Does that sound okay to you?
Yeah, will you still send me the video of the notes that we have to make sure that everything written down?
The video that we just made today?
Yeah. It didn't let me
Yeah.
see it.
It might take me a day or two to get it posted, but I will. I'll have it for you.
All right. Thank you.
Okay. I'll maybe see you Thursday. You let me know if you need anything.
All right. I should be there Thursday, so if it looks like I will.
Okay.
All right.
Bye.
See y'all.